So, a very good morning, class. Today, we are going to start with the structure of proteins and what is protein and what are the monomers of protein. That already I have explained. Yes, hope it is clear. So, amino acid is the monomer of protein, or we can say that the protein is a polymer of amino acids. When lots of amino acids come together by a specific bond, then it is called protein. And which type of bond it forms? That is called peptide bond, which is also a amide bond. Okay. You have done in polymers that amide linkage, ester linkage. So this can be a condensation polymer which is having a amide linkage, amide bond. Okay. So proteins are the polymer of amino acid joined together by peptide bond. That is called amide bond. And how it forms, that let me explain you. So this is the amino acid. I have explained that at one end it will be NH2, another end it will be COOH. In between, this number of carbon can change. It can be 1, it can be 2, 3, whatever number of accordingly. We name it alpha, beta, gamma, delta. Yes. So this is another one. So what will happen? For OH of one amino acid will react with the H of another amino acid to form water. It comes out as a water, and what will happen then? C double bond O and NH. There will be the linkage of C double bond O and H, which is also called amide. This is also called amide linkage or amide bond. And in proteins, especially this amide bond is named as peptide bond. Hope it is clear. The formation of peptide bond is not peptidation. As yesterday also I have explained that don't be confused in that word peptidation and peptide bond formation. Peptidation is in surface. That is the process to convert the precipitate into colloidal solution. But peptide bond formation, that is different thing. It is present between amino acid, because students get confused so badly in this. Yes. So now proteins are the, we can say that polypeptide, because in one protein, as it is a macromolecule, bigger molecule, so obviously so many amino acid will be there and so many peptide linkages will be there. So that's why it is also called polypeptide from amino acid. Okay. Zwitter's iron. This is again a new definition, like an important definition from this topic. It can be asked. Zwitter ion can be asked in amino acid, in biomolecules also, and as well as in aniline chapter, like nitrogen containing organic compound. There also we have done Zwitter's ion, and we are very well familiar with this Zwitter ion, that if both the cation and anion are present in the same compound and in the stable condition, then it is called Zwitter's ion. Indirectly, in general, क्या होता है, whenever this positive and negative ion are there, so they can start to, like, they accept or gain something, like that they gain or lose something to be like neutral, to be stable. But in Zwitter's ions, if you check, Zwitter's ion is stable, although it is having positive and negative ions. Got it? So due to this only, amino acid are soluble in water, because it behaves as a salt, and due to formation of peptide bond. Got it? So due to formation of Zwitter's ion. And how it forms, let me explain you. This is the amino acid. Okay, so what will happen? H, which is attached with the oxygen, being better living group, it will come out, and where it will go? Here this nitrogen is having lone pair of electron, isn't it? So this with this electron density is here high, and due to this what happened? Yes, so this hydrogen comes here and it forms ammonium NH3 plus ion. Yes, and O minus. So in the same salt, in the same compound, both the positive and the negative charge are there, and stable condition. So that is called Zwitter ion or Zwitter's ion. Got it? Both you can say. I hope it is clear. Yes. Here another thing I'm going to explain. What is this? It is see, when the pH will be low, then it will form NH3 ion over here, like this. This is the Zwitter ion. Be careful. This is Zwitter ion. Okay. where the pH is neutral. This pH is called neutral because positive and negative both the charge are there, so that's why it is neutral, and it is at neutral pH, that means around 7 pm. But when we talk about the low pH, that means less than 7, then what will happen, this becomes the acidic, and here it will be NH3 plus, but it is COOH compound. But if we make it as a high pH, that means in the basic condition, then this will have the lone pair to show its basicity, and it will lose its H plus. So it will be in the form of ion. Got it? So this is a very small thing, but if we see, it is an important point also. And on this basis, we can classify the things. Okay. So at low temperature, at low pressure pH, this is acidic COOH group will be there, and the cation NH3 will be there. But at high pH, it will be basic and it will have COO minus. Got it? Okay, so now the next thing is classification of proteins. Proteins are classified under various category. That means in general, that are the 4 category we have to learn. So first we will do on the basis of molecular shape protein. Okay, molecular shape proteins are of 2 type. One is called globular, another is fibrous, and another is globular. Okay, so first is fibrous over here. Fibrous, that means it seems as a fiber. Okay, when 2 polypeptide chain are parallel, 2 polypeptide chain are parallel with each other and attached with the hydrogen bond and disulfide bond. This is like one fiber is like this, polypeptide chain, another is this. So they two are attached with the hydrogen bond. Let me explain this hydrogen bond. Yes. And the disulfide bond will be there. Got it? Disulfide bond will be there. So these are insoluble in water. Due to this hydrogen bond, it should be soluble. But actually, here it is disulfide bond, and that is prompt over here, due to which it becomes the insoluble in water. Okay, so fibrous is insoluble in water. And example like keratin. Keratin is present in our hair. Yes, wool, silk, myosin which is present in our muscles. So that's the reason that keratin. Otherwise, if we will like if it is soluble in water, so all the protein will be soluble in water. That keratin here, the protein which is present in here, that will be soluble, and at last we will not have any hair, proper hair. Yes, because all that protein is dissolved now. So it will be like we can say that the hair structure. Although I will explain all the things कि क्या होता है, why the hair becomes rough like this. Yes, जो बाल rough हो जाते हैं, why so that I will explain later on. Okay. So now next is globular protein. Globular protein is like when it is coiled or coil होके ये spherical shape में आ जाता है, then it is called globular protein. Okay, when polypeptide chains coil around to give a spherical shape, then it is termed as globular protein. And these are soluble in water. Example, insulin and albumin. Okay, albumin, where you have seen that the egg white has albumin protein. Okay, so insulin, you all know that it is. So these are the on the basis of molecular shape, they are of 2 types. Okay, we can see that another one, it is collagen, and this is the polypeptide chain, how they are parallel to each other, like they are helically structured. And in this case, it is hemoglobin. Hemoglobin may we have iron, which is also known as this is the hemoglobin is also a protein. Got it? So this type of things, the structure are there. Like now, different types of protein on the basis of types of bond. Which type of bond are present between the proteins? That on that basis, the proteins are again classified, and they are of 4 types. First is primary structure. Primary structure का मतलब it will have a like straight, it will not be coiled. Okay, and only that if may have one or more polypeptide chain, obviously. Okay, amino acid linked with each other in a specific sequence. एक specific sequence होगा. How? Let me show you. Like this, you can see this is NH again here. that is the carbon chain, alkyl group, and then CO NH bond, amide bond over here, again the amide bond over here. So it will be a straight chain, एक straight pattern में होगा, ठीक है, एक specific sequence में होगा. Like all these are the amino acid, tryptophan, you can see that tyrosine. Before, let me show you again. This is cysteine, देखो glutamine, arginine, methionine, all that are essential and non-essential amino acids are there, and they are going to attach with the, you can see trypto here, like valine, leucine, tyrosine, isn't it? So all this structure will arrange in a specific sequence and will form a straight chain like this. Okay. Now, secondary structure of protein. Secondary structure of protein, that means it will be little coiled, okay? And again, the secondary structure of protein is divided is of two types. They exist in two different types, alpha helix and beta pleated sheet. Okay, due to folding of, sorry, it's only CO, okay? It's not COO, it's CO and H2 bond. So due to the folding of amide bond, there will be folding of amide bond and intermolecular hydrogen bond की वजह से it will have two type of structure like alpha helix and beta pleated sheet. Okay, got it? So if we check that in this case, like it is here, it is alpha helix, it will be in form of helix. Okay, like एक spiral, एक imaginary line के around it will be spirally coiled. Okay, and this is pleated sheet, it will be a sheet like structure. So this is that 3D structure I have shown to you. So you can see that how the bond of how the CONH bond is going to be folded. Yes, this is, you can see this is lighter and darker, that means ये front में तो ये back में, so there is a folding in that. That's why it gives the two type of structure. One is if hydrogen bond की भी folding हो जाए तो it can give you that alpha helix also, and here it can give beta pleated. So why it happens, why this gives two type of structure? Because of folding of CONH2 bond and intermolecular hydrogen bonding. Got it? समझ में आ गई बात? Okay, so now next thing. This is the primary structure it was. Now it is going to be little coiled, okay? Or thoda coiled ho gaya, and this is going to give you secondary structure. Yes, this is the primary structure. Now it is going to be little coiled, and that is going to arise that formation of secondary structure. You can see that the secondary structure like this, how it is going to coil. This is alpha helix, yes, and this is the folding of bond, this is side chain, and secondary structure when it is beta pleated, how it will form. You can see that the alpha helix only I have that diagram, so you can see that. Yes, now the tertiary structure of protein, the third type of structure. यहाँ पे क्या होगा, amino acid protein chain will coil more, okay, will be coiled more. So overall folding of polypeptide chain will be there, like highly coiled, you can say. Forces present between this tertiary structure of protein will be hydrogen bond, disulfide linkage, van der Waals force of attraction, and electrostatic force of attraction. In the secondary protein, it was only hydrogen bond and amide bond, peptide linkage. In the tertiary structure, it will be hydrogen bond, disulfide linkage, van der Waals force of attraction, and electrostatic force of attraction. All four will be there. So sometimes it is asked that which type of force of attraction are present in tertiary structure, in secondary structure like this, they can ask. So you have to learn this point, that van der Waals force of attraction, disulfide bond, hydrogen bond, and electrostatic force of attraction are present in the tertiary structure. So you can see that this is a disulfide bond. Yes, sulfur bond will be there. So you can see that it is hydrogen bond. Here it will be types of side chain interaction, how this side chain is going to interact. So you can see that I said that electrostatic force of attraction, that means ionic bond. Here it is disulfide bond. Here it is hydrophobic interaction, that means it is going to make a chain of carbon, and that will be the hydrophobic, that means that will not dissolve in the water. Yes, and this is hydrogen bonding. So this step, and it will lead to this type of structure, a 3D structure, that means so coiled. Okay, you can see that earlier it is coiled inside, and then this one is also coiled. So it is coiled, little coiled. Okay, little more coiled. Okay. Now you can see that this was the secondary protein, and now it is going to be coiled again and again, and this will form the tertiary structure. I hope it is clear. Yes, secondary protein, secondary structure of protein, and now it is tertiary structure of protein. Okay, so now the last type of last structure of protein, that is quaternary structure of protein. Quaternary is highly coiled. Okay, too much coiling will be there, and it's highly coiled. You can see that how it is coiled. You didn't get the terminals or the सारे कुछ किधर से कौन सा mix हो रहा है, you can't see. Yes, so this is alpha chain, beta chain. ये सारे alpha chain है, and this is beta chain. Okay, they are again going to coil on that. This is the you can see that the diagrammatic with colorful picture, that is the quaternary protein, which is highly coiled, and all type of force of attraction is present in this. Yes. So if you see that, let's do the recap. Primary structure, let me show you again. This is the primary structure. You can see that the single, yes, no coiling. Secondary structure, little coiled. This is alpha helix, and this is the beta plated. Tertiary structure, coiled. Isme alkyl group interaction will be there, and the quaternary structure, highly coiled. Proteins with more than one polypeptide subunit. Okay. So the now topic is denaturation of protein, and as the name suggests, something we are going to do with its native structure, native nature, like nature. Yes. So denaturation of protein means that if we subject the protein to any changes like pH changes or something temperature changes, then the structure of protein gets denatured. That means the structure of protein gets changed. Okay, so that is called, the nature of the protein gets changed, and that is called denaturation of protein. Hope it is clear. So the protein found in the nature is termed as native protein. When it is subjected to physical change like temperature or chemical change like pH, either हम temperature change करें या फिर pH change करें, then their hydrogen bond get disturbed, or not only hydrogen bond, rest all the bond can be they get disturbed, and due to which they lose its native structure. Got it? And that is called denaturation of protein. Due to which globules unfold and helix get uncoiled. अगर ग्लोबुलर प्रोटीन है तो वो चूंकि बहुत coiled होते हैं, they spherical shape में, they gets unfold, and helix structure जो होता है, they get uncoiled, and the protein loses its natural activity, and that is known as denaturation of protein. Okay, so this is again a important set of question. Can be asked that what is denaturation of protein? So denaturation of protein, you have to say that when the protein is subjected to physical change like temperature and chemical change like pH, their hydrogen bond get disturbed, due to which globules unfold and helix get uncoiled. Okay, and protein loses its natural activity. If you are not clear with this, ये वाला point अगर आपको यहाँ पे not clear, globules unfold and helix get uncoiled, you can leave this point. Okay, you can simply write that, and protein loses its natural activity, due to which that is known as denaturation of protein. Okay, due to denaturation of protein, secondary and tertiary structure are destroyed, but even quaternary also, क्योंकि coiling वहीं पे है, but primary तो uncoiled है, isn't it? Primary में कोई changes नहीं होगा. So that's why secondary, tertiary and quaternary structure will be destroyed, but primary structure remains intact. Primary structure में कोई changes नहीं आएंगे, क्योंकि उसमें कोई और bond है ही नहीं. It's a simple peptide bond only, isn't it? Okay, so example, coagulation of egg white on boiling. When we boil that egg, it gets like hard हो जाता है थोड़ा, and that is coagulation of egg white. We say that is also an example of denaturation. Curdling of milk का जमना due to formation of lactic acid by the bacteria in the milk. Got it? So that is called denaturation of protein. So our next topic is enzyme, but before that, one macromolecule we have not covered, and that is not so important actually, so that's why we have not covered here, and that is lipid. Okay, so lipid gets emulsification जब it's emulsified, and it gives smaller molecules of lipid, and then lipase enzyme work on that, and it gives like fatty acid and glycerol. That is something bio. So we are not concerned with this. So let it be. Now let me start with this enzymes. So enzymes are also a protein. Actually, it is made up of protein. Got it? And it works on the macromolecules, all the type of macromolecules, and break that into the simpler form. Got it? So enzymes is a protein. Actually, it is made up of protein, which works on every macromolecules and split them into a smaller molecule. Got it? So let's see. Such biological compound which coordinates in the various chemical reaction in living organism. Mostly enzymes are protein. And which type of protein? Globular. Generally, they are named according to their compound substrate on which they work. And a good thing I'm going to explain about this. This enzyme will always end with ASE. Okay, I have explained about the carbohydrate. Carbohydrate ends with O's. Protein ends with EIN or IN. And enzymes end with A's. Got it? So, it is named generally it is named on according to the compound or the substrate on which it work. Like if it is working on lipid, its name will be lipase. Yes, if it is working on amylose, what will be the name? Amylase. If it is working on urease. If it is working on sucrose को हम invert sugar कहते हैं. Okay, we will not say sucrase. Enzyme is not sucrase. So sucrose name जो है, that is also called invert sugar. So from there its enzyme name we can find, and that is invertase. Okay. Similarly, glucose ko the enzyme which works on glucose is not named as glucase, and instead of that it is named as zymase. Zymase, ZYMASE. MAS, yes. That is the enzyme which work on glucose. Okay. So rest is important, very easy. You can find with this. Okay, only the zymase that work on glucose, and invertase which work on that is invert sugar, that means sucrose. Rest you can easily find. Okay. So sometimes they named after the reaction in which it involves, like oxidoreductase, where you have seen all the bio students must be knowing this oxidoreductase in the what we say Krebs cycle. Yes. So there you have learned, I think, third or fourth step it is oxidoreductase enzyme is used, which means oxidize, what it does do, it oxidizes and one as well as reduces the other. Oxidation reduction reaction वहाँ पे करता है this enzyme, and that's why it is named as oxidoreductase. So sometimes it is named according to its work, what it is working, and sometimes it is named according to the compound or substrate on which they work. Okay. So example, maltase enzyme will work, lactase enzyme will work. You can write for this zymase, which work on glucose, and invertase. This is invertase, which work on invert sugar. That means sucrose. Invertase, which work on sucrose. And why it is named like? Because sucrose is known as invert sugar. Okay, what is the mechanism of enzyme action that we have already done in chemistry and everyday life? But let me repeat it again, that suppose that this is the enzyme, this work on the lock and key concept. Okay, so that means for particular lock there is a particular key. Similarly, for particular substrate there is a particular enzyme. Okay, or you can say that the particular enzyme will work upon the particular substrate. So like this is the enzyme, and the site, all the site where the substrate attaches with this enzyme is known as active site, and other sites than site other than the active site is known as allosteric site. You can write, you have done only this thing we have done in what chemistry in everyday life? Allosteric site. Other than active site is known as allosteric site. Okay. So, and then when this substrate is going to act on this, it is forming enzyme substrate complex, and then there will be reaction, and this reaction is going to energy is absorbed, bond will be formed here with the substrate के बीच में bond बन जाएगा, and then what will happen? This will come as a product outside, and that enzyme is again free to form another bond. Got it? So this is the enzyme action. This is the mechanism of enzyme action. This is a very funny GIF. We can see that enzyme, how it is going to, that substrate, a particular like enzyme is there with a particular active site, and that substrate is going to attach. So I found it very funny. That's why I have added it here. Now again, next GIF, you can see that how it is going to act. Okay, on which site it is acting? Reactive site or active site. Other sites are called allosteric site. Okay. So now this is vitamins, another micromolecules, okay, and we need this in very small amount, in very small quantity we need all these vitamins. So we know that there are two types of vitamins, in we have done in 7 or 8 something. Yes, such compounds which are required in very small quantity for normal maintenance and growth of organism is known as vitamins, that are also called micromolecules. Okay, plants can synthesize all types of vitamins. That's why कहा जाता है कि green vegetables, leafy vegetables खाइए आप, because plants और vegetables में हर तरह की vitamins होती है. Got it? Instead of taking multivitamin, we can eat like all that green leafy vegetables or all that vegetables. We will get the vitamin in sufficient amount. Okay. So vitamins are of two type. They are of two type: fat soluble, adic. To learn, you can say that adic. Vitamin A, vitamin D, vitamin E, vitamin K. These are the fat soluble. That means they can be stored in our body, and where? In the adipose tissue. Adipose tissue, bio students must be knowing this thing, where it lies. It lies just below the skin. Yes. So ADEK, that can be stored in our body, stored in our liver or adipose tissue. जहाँ body के जिस part में adipose tissue है, that it can be stored. And another is water soluble. So from this topic, two type of question will be asked, that define vitamins and what are the types of vitamins, like this. Okay. Second question, what are the fat soluble vitamin? So, or what are the water soluble vitamin? It will be asked. And third question, which is the most frequently asked question, I'm telling you, water soluble vitamin जो होते हैं, that is rest other than this adic, यानी कि B और C बच गए. So B और C vitamin जो होते हैं, they are water soluble, and since they are water soluble, so that's why they can be excreted in the form of urine, except B12. B12 हमारे body में मतलब स्टोर रह जाता है. रेस्ट ऑल दैट विटामिन बी एंड सी, जितने भी विटामिन के विटामिन के बहुत सारे पार्ट्स होते हैं, विटामिन 1, विटामिन B1, B2, B3, B4, something like that. So for every particular vitamin B1 or B2, there is a problem, there is a deficiency disease that we have to learn. So all that vitamin are water soluble, that's why they can be excreted in the urine, except vitamin B12. Okay, and therefore these are essential in our diet, and that's why we are supposed to take in the regular basis in our diet. Now the question, important question which will be asked, that why vitamin C is required in our daily diet. ठीक है, और विटामिन बी भी पूछा जा सकता है, बट फ्रिक्वेंटली आज जो है वो विटामिन सी है. वाई विटामिन सी इज required in our daily diet? Because vitamin C is water soluble, so that's why it can be excreted in the form of urine, and hence it is essential in our diet. I hope it is clear. So this is the important question from this topic. Okay, now this is the table you have to learn. Bio students has to, like, it is must for them, they have to learn. But for the chemistry, very few questions will be asked, like deficiency of vitamin A, what it causes. Okay, vitamin B2 or vitamin B simply पूछा जाता है, vitamin B की deficiency से. So we will consider that it this B1 consider करते हैं. If it is B only पूछा जाता है, तो we will consider that B1, and very is the problem. Okay, दैट मीन्स लॉस ऑफ एपेटाइट, भूख नहीं लगती है, जिसकी वजह से ग्रोथ रिटार्ड हो जाती है. एंड अगेन ये क्वेश्चन पूछा हुआ है बहुत बार, what is the deficiency disease caused by vitamin B12? That is pernicious anemia. That means RBC deficient in hemoglobin. RBC डिफिशिएंट हो जाता है, हीमोग्लोबिन का कलर जो रेड कलर जो RBC की वजह से होता है, एंड दैट्स वाई इसके कलर में कुछ थोड़ी सी प्रॉब्लम होती है, और दैट्स वाई वी से दैट कि येलो बॉडी होने लगती है एंड एनीमिक हम बोलते हैं. दिस इज पर्निशियस एनीमिया. ओके, विटामिन सी इज आस्क्ड, विटामिन B12 and vitamin C is mostly asked question. Okay, these are like, let me triple tick. Okay, so vitamin C जो होता है that is present in our all that citrus fruit, जितने भी हमारे खट्टे फ्रूट होते हैं, green leafy vegetable, सब में विटामिन सी होता है, एंड इट इज एक्चुअली विटामिन सी इज एस्कॉर्बिक एसिड. ओके, एंड इट लीड्स टू स्कर्वी. स्कर्वी मीन्स ब्लीडिंग गम्स, गम्स में ब्लीडिंग होने लगती है. सो क्वेश्चन पूछा जाता है व्हाट इज द डिफिशिएंसी डिजीज ऑफ विटामिन सी, सो दैट यू हैव टू लर्न. Rest are important but less asked. Okay, विटामिन ई दैट इज ड्यू टू दिस इंक्रीज फैसिलिटी ऑफ RBC एंड मस्कुलर वीकनेस हो जाता है, मतलब बहुत ही. विटामिन के की वजह से इंक्रीज ब्लड क्लॉटिंग टाइम, मतलब ब्लड क्लॉटिंग नहीं होती है. सो दिस इज, एंड विटामिन ए की वजह से जीरोप्थैल्मिया, यू आर सपोज टू से नाइट ब्लाइंडनेस भी होता है, but being classed like it is importantly asked, like it is frequently asked xerophthalmia. So you are supposed to answer in form of xerophthalmia. Okay, what does it what is the problem in this? This is hardening of cornea of eye. So that is the few important vitamins. You have to learn that what is this, where it can be found, what are the sources, and what is the deficiency disease for that. So these are the important thing, and next thing that we will do in our next class. So till then, thank you class, enjoy your study.